Hello. Today I'll be talking about DNA supercoiling. I'll be essentially giving an overview of this review paper by Ivan Jr. and hopefully this will give an overview to someone who hasn't been exposed to DNA supercoiling at all. So this is the outline of the talk. I'll first start with describing what supercoiling is, and then I'll move to a more biological context of why supercoiling occurs in bacteria and cells. And finally, I'll talk about what the consequences of supercoiled DNA are on the structure and other functionality of DNA present in bacteria and cells. Let's first start with the basic concepts of DNA structure. So here I've shown the diagrammatic structure of a double helix DNA strand. It has these sugar phosphate backbones on either side, and the two strands are connected via nucleotide bases, as shown here. This is the planar representation of the molecule. And in this, if the DNA were to take this structure, then as you can see, there is space between two consecutive layers, pairs of nucleotide bases here. But people have found that DNA exists in this double helix turned sort of structure. So the reason why DNA prefers to be in this double helix turned structure as compared to this parallel planar structure is something to do with the structural chemistry of the molecules involved. Each of these, the molecules on the backbone are hydrophilic, which means that they like water, whereas the nucleotide bases, the ones inside, are hydrophobic. And since the DNA is present in an aqueous environment usually, if the DNA were present in this open parallel planar structure, then water molecules could get inside the spaces between nucleotide bases, which would be energetically unfavorable. In order to avoid this, one of the ways that DNA can structure itself is to minimize the space between these bases. So again, I've shown the same thing here. Each this DNA strand are shown in yellow here, the backbone, and the bases are shown as a cuboidal box. So one of the ways that these boxes can be stacked on top of one another is in such a way, such that each box is slightly tilted with respect to the box below it. And what this achieves is that there's no space left between these two boxes. So no water molecules can get in, and water molecules can stay outside where they're in contact with the backbone only. If we continue this pattern by stacking more and more nucleotide bases, eventually you'll see the double helix being formed, the whole helix turning. Something like this. Yeah. In normal circumstances, this turn is well defined. So each one single 360-degree turn that the backbone takes roughly consists of 10.5 of these base pairs. And this is in normal conditions in an aqueous environment. However, if for some reason the DNA is underwound or overwound or undertwisted or overtwisted, then this number of base pairs that constitutes one single turn can change. It can be either undertwisted, which means that the number of bases is higher, or it can be overtwisted, which means that one single turn has lower number of bases. And this sort of state is what leads to supercoiled DNA. This is a local twist property, which eventually leads to non-local effects, such as shown here. So for example, here is a telephone cord, which is itself a coil. And because of the turns imposed on the wire, you can see that there's multiple of these protruding structures that form. This is essentially what supercoiling is. Because of the intrinsic twist of present in the DNA, it takes higher order supercoiled structures in cells. And uh, this is what we call as a supercoil. It can have many various forms, either toroidal or plectronemic, and people have mainly tried and observed these plectronemic structures in cells. So how does one quantify supercoiling? Like I said, there's two properties of supercoiling. One is local and one where it's non-local. The local quantity is called twist, and it is defined as the number of 360-degree turns, integer 360-degree turns, that is imposed on a straight filament. So here, this description is general. It can apply to any two parallel curves. It doesn't have to be DNA itself. And once the twist is introduced and the ends are fixed such that they cannot rotate, then this twist is locked into the system. And we can describe this twist by the number of 360-degree turns. It has a sign convention depending on which way the two curves are twisted. And this will be important when we also consider the non-local effects of supercoiling. The non-local effect is quantified by something called the ride. It is the number of times the whole axis of the two curves crosses itself. As I showed earlier, in supercoil structures, because the entire axis is crossing over itself multiple times, this sort of quantity gives the non-local quantification of supercoiling. Again, this quantity also has a sign convention, and the sign convention is defined such that it doesn't matter from which projection the entire molecule is looked at. So for example, here, the it shows one crossing in this diagram, and the top strand moves in this way from left to right, whereas the bottom strand moves from the left of the top strand to the right of the top strand. So this is defined as negative supercoiling, and the opposite is true for positive supercoiling, when the bottom strand goes from the right of the above strand to the left, and that is defined as positive supercoiling. This might be confusing because here the direction of the top strand is reversed. So the right is actually on this side and the left is on this side. Here I shows two examples of negative right and positive right. And when there's multiple crossings, then right can also take higher values. In the case of DNA, the twist and the right are usually always in integer values, although for a more general setting, this need not be true. One of the ways that we can quantify supercoiling very well is via the linking number. And here you may have noticed that this twisting and rising of the two curves can be interchanged. Even in the telephone cord that I showed above, it is only because the local twist gets converted to right that the cord takes its supercoiled structure. So how this occurs is that there is a topological constraint between any two curves that we can impose, which is called the linking number. In such topological constraints, that is when the two ends of the curves are fixed such that they cannot rotate, or when they are closed, when the ends of the two curves are closed in a loop like this, then the linking number of the two curves is always conserved. It cannot change. The linking number is essentially the total number of times that the two curves cross in 3D space. So it's a combination of the local twist, which indicates the crossing of the two curves locally, and the right, which indicates the crossing of the axis of the two curves. And so the sum gives the linking number, which remains conserved when topological constraints are met. For example, here, there's one twist imposed in these two circular curves, and the linking number is calculated to be 1. This diagram again shows how the twist and writhe can interconvert between one another. So here in this diagram, there's a total twist of +3 in a rubber tube that is taken, and this can get converted to different forms, different values of twist and writhe, until eventually all the twist is converted to +3 writhe. This sort of interplay between twist and writhe is dependent on the energy of either twisting or bending. Usually in such rubber tubes, the energy of bending is lower as compared to the energy of twisting. So the ring prefers to be in a writhed state rather than a twisted state. Now coming to the confirmation of this sort of conversion between twist and writhe, it has been observed in simulations as well, simulations that have tried to model supercoiled DNA. Here they have chosen a simple model of a rod-like chain where they have taken a polymer, and each unit of the polymer is a rod which has a certain torsion angle, and also between consecutive rods they have defined a bending angle. Based on these two angles, they can calculate the energy of torsion and bending based on the torsion and bending rigidities defined for this polymer. And once they impose an amount of twist, intrinsic twist that is present in the system, they can observe these sort of supercoiled pectoneme structures, which confirms what we also see in supercoiled DNA in cells and bacteria. So one way to quantify the supercoiling amount of supercoiling present in the entire system is via the supercoiling density. It is defined as the ratio of the excess linking number divided by the linking number present in the system in a relaxed state. So for example, if I take a bacterial plasmid, which is a circular chromosome, and in normal conditions, each turn, each 360-degree turn of the DNA helix consists of 10.5 base pairs. If the plasmid is 1050 base pairs long, then we can calculate that the linking number, or the number of 360-degree turns, in this case, only the twist, would be 100. Now, if we suppose impose an additional twist by doing an operation on the circular chromosome, so we impose one additional right-handed twist so our excess linking number becomes +1. Now, the supercoiling density, which is defined by this ratio, becomes 0.01 with a positive sign. This sort of supercoiling density is what is reported when people study supercoiled DNA. And in mesophilic bacteria, which is some kind of bacteria, the chromosome or the DNA is on average negatively supercoiled with a value of -0.05. The negative here just implies that the DNA is slightly unwound on average. And I'll make it clear how this occurs, what causes the unwinding of DNA in subsequent slides. Just a brief overview here of how people actually measure supercoiling or observe supercoiling. One of the most standard techniques is to use gel electrophoresis, where people take an agarose or some kind of gel which has small pores in it. And once they place DNA on top of the gel, it can permeate through the holes present in the gel. And the size of the DNA determines the mobility of the DNA. When they apply an electric field from on either end of the gel, because the DNA is charged, it prefers to move in a certain direction. And we can measure the mobility of this DNA, which can give an estimate of its size. If it's more supercoiled, then it tends to take a compact structure, so it moves more in a short amount of time, as compared to when it's not supercoiled, where it has a sort of spread out structure. Another way is to actually perform electron microscopy, which can give us the rigid structure of the DNA directly, as shown here. And then there are also molecules called intercalators, which can specifically bind to regions of lower or higher supercoiling, and which can be then imaged via fluorescence microscopy. Now I'll switch to a more biological context of why supercoiling occurs in bacteria. Before I go to the agents that cause the supercoiling, first let's establish how the DNA is constrained topologically, such that supercoiling can actually occur. So there have there have been debates about the exact mechanism of these topological constraints, but some of them include the transcription machinery. So during the transcription of DNA, there's a protein or an enzyme called the RNA polymerase, which can bind on top of the DNA and move along it while transcribing an mRNA transcript, which just copies the information present in the DNA into an mRNA transcript that can then go and form proteins. When this polymerase binds, it can act as a topological constraint, where locally it does not let the the bound DNA rotate because of this bulky machinery that is attached to it. Another way that the topological constraint can be imposed is via other proteins called DNA bridges, a class of proteins called DNA bridges, which can bind two different points of the DNA contour like this. And because it binds it in such a way, the total twist of the loop that it confines, it cannot escape to other parts of the DNA contour. And so if there is some change in the linking number in this part, it stays there and it gets supercoiled. So one of the agents that introduces supercoiling is again this RNA polymerase during the process of transcription. Here I'll just briefly describe how transcription occurs. When this RNA polymerase binds to the DNA, it first unwinds it at the starting end, and it such that the two strands of the DNA are separated. Then it can access the one of the template strands of the DNA and create an mRNA transcript, which can then be used to create proteins. As it moves along, the RNA polymerase rezips the two strands of the DNA back to form a double helix again. Now, because the DNA is already in a wound structure, even in its natural state, as the RNA polymerase moves along the contour of the DNA, it must first unwind the two strands of the DNA so that it can separate them. However, because the RNA polymerase is sort of bulky, it forms a complex with lots of many other proteins, and so it has difficulty in rotating along the contour of the DNA. So, as a consequence, what can happen is that the RNA polymerase, which is indicated by this blue triangle here, it just moves along the DNA contour without rotating. And as a result, the two strands that it splits in its upstream region, they stay sort of in this planar conformation, like this, in their underwound state. And because of this, because there is no net supercoiling or linking number being imposed in the system, the topological constraints enforce that the linking number must be conserved. And so the excess winds that were lost here in the upstream region get transferred to this downstream region, which gets more and more supercoiled. So there's a positive supercoiling here and a negative supercoiling here. And this happens essentially because the transcription machinery or the polymerase cannot rotate. It faces torsional friction. So here I've um, tried to find a video clip that can explain, that can show this process happening in action. Here they have taken a rope that is again wound in a double helix to model the DNA. And this hand is supposed to represent the transcription machinery. It will first open a strand or a region of strands here by unwinding it. And because the strands are unwound here, excess windings will appear in this region here. So here, this hand unwinds the strands, and because of that, there's a bubble of unwound strands that form here, where the RNA polymerase can access the template and form an mRNA transcript. But if you look closely in this region, the extra windings that were lost here were transported here. So this region is actually now overwound. I'll try and play this again so that you can focus on this region this time. Here, as he unwinds this region, the windings here increase. And this is exactly what happens in the supercoiled state during transcription as well. So here, again, I've shown the same thing. Ahead of the polymerase in the downstream region, the DNA gets positively supercoiled, whereas in the upstream region, it gets negatively supercoiled. There's no net change in the supercoiling over the entire chromosome, just this local positive and negative supercoiling on either side. Hmm. This sort of positive and negative supercoiling can actually pose a challenge for the polymerase to continue its transcription activity. Because if the downstream region gets too overwound, it gets very tightly coiled, then the polymerase will stall. It won't be able to unwind the DNA any further. So in order to avoid this, in order to avoid the polymerase stalling, there are other proteins that can help release this overwindedness of the DNA or underwoundedness of the DNA. These proteins are called topoisomerases, and they are divided into two classes. Class 1 topoisomerases, they take individual strands of the DNA and make them pass through each other, like this, as shown here. So here they've first shown that the two strands are slightly unwound, and because of the activity of this topoisomerase, it makes the orange strand pass through the green strand and introduces an extra winding. So this essentially introduces positive supercoiling, which can be helpful to release the negative supercoiling in the upstream region of the transcription polymerase. In the class 2 polymerase case, the two, it can actually, the protein can actually completely uh, make the two independent double helices cross each other, as shown here. So here they have shown first a single a double helix, one particular double helix of DNA, and here is another independent double helix of DNA, which could have actually formed a loop like this. This protein acts such that it first splits the one of the helices of the DNA, and then makes the other go through it and rejoins it back, such that now the two double helices have entirely crossed. What this does is that it can change the rise of the entire chromosome by +2 or -2. And again, so this can help relieve the positive supercoiling that builds up in the downstream region of the DNA. Just to mention, it is the action of these topoisomerases which can actually change the linking number uh, that affects the total supercoiling density present in the entire chromosome. So it is the effect of these topoisomerases as well as processes such as transcription and replication, which affect the supercoiling density present on average over the entire chromosome. Jumping to replication, in a similar way to transcription, this can also introduce supercoiling. Replication is when uh, the two strands of the DNA are first split into single strands, and then each strand is replicated so that it creates its own double helix in the upstream region. This is done by another replication protein, and in a similar way to transcription machinery, it first opens up the double helix DNA strand. So in the same way, because this replication machinery is bulky, it faces torsional friction, and so as it's moving along the DNA contour, it introduces overbindings in the downstream region, whereas the DNA is relatively the two strands of the DNA that are replicated are relatively underwound in the upstream region. So this sort of introduction of positive supercoiling is the exact same mechanism that I discussed in the transcription case as well. However, there is one other thing that can happen special in replication. Over long time scales, this replication machinery can actually rotate. It's just very slow. And when it does rotate, what can happen is that because it's replicating two individual strands into full double helices, the replicated DNA helices actually get wound together on their in the upstream region, like this, as shown here. So what this means is that each of these individual thick lines is a double helix itself, and it forms a super helix with another DNA double helix strand. This can pose a problem because usually when DNA is replicated, it is during the cell division cycle, and so the two copies of the DNA must be segregated to two different parts of the cell so that the cell can divide. However, if the two copies are wound together like this, then it poses a challenge to segregate to two different parts. So again, the topoisomerase proteins come in to help resolve these kinds of findings. Especially, there's a class 2 topoisomerase called topo4, which helps resolve such windings such that the two DNA copies can move apart, can segregate. Now I'll jump to consequences of supercoiling. We have seen that mainly transcription and replication can impose these supercoiled structures, and the action of topoisomerases can influence the amount of supercoiling, in some cases release the torsional constraints, which otherwise would lead to stalling of transcription or replication processes. One of the ways that the supercoiled DNA helps in certain bacteria is that it helps the DNA or the chromosome compact itself further, which can help form the nucleoid. So here I've shown an illustration of a E. coli bacteria, and this yellow stringy substance is the DNA or chromosome content. It occupies a subvolume present in the bacteria, which is called the nucleoid. And in order to be packed into this very compact structure, one of the aids could be supercoiling. So here I've shown a ring polymer, which represents the ring chromosome of a bacterium. And in polymer physics, it is established that the size or the radius of gyration of such a ring polymer scales as n to the power nu, where n is the number of units present in this polymer, and nu is the scaling size factor, which is close to 0.6 for these kinds of rings. However, in the case of supercoiling, when the local DNA is twisted due to the action of various proteins that I discussed earlier, it can form this sort of supercoiled branch structure, which is more compact. Such a branch polymer has a nu of 0.5, which is less than that of the ring. So that means that the size of this branch structure is lower as compared to the ring. And because of this compaction, it can help the DNA more easily attain the nucleoid conformation. Another consequence of such supercoiling is in the form of transcription bursts. So, what transcription bursts are is that people have observed in cells when certain proteins are transcribed, they are transcribed in a large amount. Whereas when they are not transcribed, uh, there are periods where they're not transcribed at all, followed by periods where they're transcribed in a large amount. So one of the ways to understand this is via supercoiling. As I explained earlier, RNA polymerase, it unwinds the DNA strands in order to access the template. And this sort of unwinding, as is obvious, is easier to do when the DNA is already unwound, when it's already in a negatively supercoiled state. I also explained that this RNA polymerase induces a region of positive supercoiling ahead in the downstream region and a region of negative supercoiling in the upstream region. So what can happen is that one of these polymerase can first bind to the DNA transcript and it can move along the DNA transcript while making the mRNA and introducing these opposite signed regions of supercoiling. Because the upstream region, the DNA is slightly underwound because of the action of these polymerase. Another RNA polymerase can bind, and it can bind more easily because it's already unwound, and it can again uh, it can go along the transcript, introducing a region of negative supercoiling behind it. So if there's more of these RNA polymerase in the region behind them, the DNA is more negatively bound, which makes it easier for other RNA polymerase to bind in that region. And this leads to a positive feedback loop. So this can explain the transcription burst that I explained, that I briefly alluded to. When there is multiple of these DNA polymerase or RNA polymerase binding and transcribing the mRNA, lots of proteins can uh, be transcribed at once because of this positive feedback loop. But when all of them stop, when all of them detach, then almost no activity of this transcription occurs. Thus, this sort of positive feedback loop leads to bursts in transcription activity. Another consequence of supercoiling could be in the mobility of these DNA plasmids. So this was a study done by Smrek et al. In a melt of supercoiled rings of supercoiled polymers. So does not directly relate to the case of bacterial or eukaryotic plasmids, but it still might be relevant. What they showed was that as they increased the amount of supercoiling density in the ring polymers that they took, the mobility or the diffusion constant of those rings actually increased. The reason that they gave for this was that when they increased the supercoiling, the area or the volume that the rings occupy, it becomes more compact as compared to when it's open like this. What this means is that because the area is more compact, lower number of other polymers in the melt can be threaded through the area of another polymer. In the case where there's no supercoiling like this, there's more area to be threaded. because it's in an open conformation. So it's relatively easier for another polymer in the melt to just go through this area and get entangled with this polymer. Whereas in the supercoiled state, because there's lower area exposed, the threadings are more difficult and rarer. As the number of these threadings decrease when we increase the supercoiling, the this number of entanglements decrease, and that leads to an increase in mobility of the DNA plasmids. This can have implications for eukaryotic chromosomes, for example, but it's not a direct model yet. Hmm. Just to summarize the kinds of models that people have used to try and study the properties of supercoiled DNA. One class of models takes a look at the local properties and builds up the behavior at longer length scales. For example, the rod-like chain model that I talked about earlier, where they model each unit of the chromosome as a rod, and this rod can model can represent 10 to 100 base pairs of DNA length. However, these types of models are restricted in that they can only predict behavior of stretches of the chromosome that are roughly 20 kilobase pairs long at maximum, which is lower than two orders of the total chromosomal length. This is because of computational costs that are associated with simulating these kinds of models. Another way that people have tried to model the long level behavior of supercoiled DNA is to take an effective polymer approach. So because supercoiled DNA can take these sort of branch polymer structures, they are modeled as branch polymers. But another problem with this is that they lack microscopic explanation. And so they require fitting of certain parameters that characterize these kinds of structures, which can be done by data-driven analysis obtained from experiments. However, there is still a need for models that can safely predict, based on molecular microscopic explanations, the long-range behavior of such supercoiled structures. So in summary, what I've talked about is how under and over twisting of DNA leads to these supercoiled structures, which is observed in cells and bacteria. And these kinds of supercoiling in bacteria are caused by transcription, replication, and the effect of topoisomerases. As to why the supercoiling occurs is that it can pose uh, certain advantages to the bacteria or the cell. It can lead to the compaction of chromosomes to form the nucleoid in bacteria. It can also help regulate gene activity in the form of transcriptional bursts. And there are other various functions that I did not describe that people are still currently investigating because it is not very clear what the main function of such supercoiled DNA is. However, because the cell is investing so much energy, people are sure that there must be some good way that supercoiling helps in the functioning of the cell. Thank you.